This episode of Cell and Gene the podcast is brought to you in partnership with Thermo Fisher Scientific. Thermo Fisher's cell therapy processing instruments are designed to help customers transition from process development to commercial manufacturing, utilized as standalone devices or integrated as part of a closed modular process. Thermo Fisher Scientific recommends Gibco CTS DynaSelect Magnetic Separation System, which is a next-gen cell isolation and activation instrument. Gibco CTS Xenon Electroporation System allows customers full control to optimize for a variety of cell types and payloads. And Gibco CTS Rotea Counterflow Centrifugation System is a closed cell processing system supporting a broad range of protocols for cell separation, washing, and concentration. Customers can rely on and streamline their drug development process with Applied Biosystems QualTrack qPCR and dPCR quality control tools for robust and reliable genetic analysis across various phases of drug development, supported by relevant, compliant documentation. Hello, listeners, and welcome to this episode of Cell and Gene the podcast. I'm your host, Erin Harris, and my guest for this episode is Andy Ramelmeier, EVP, Head of Technical Operations at Sangamo Therapeutics, a biotech based in Brisbane, California, that applies cell and gene therapy to combat hemophilia A and other genetic diseases. So, Andy, thank you so much for your time. I'm thrilled you're here with us.
Yes, great, Erin. Great to be talking to you.
Absolutely. So, all right, let's jump right in. Now, a few years ago, Sangamo built its own phase 1/2 cGMP AAV in-house manufacturing facility. So that's a big deal. And we want to break this down as to, the why behind it, the successes, all of that. Let's start off with this though. What drove the decision to build, say, versus buy?
Well, a lot of that just comes due to the complexity of our products. Now, I'll talk about AAV, but we also have cellular products as well, and they're very difficult to make. And we've found that we have better control of cost, schedule, and quality when you have your own facility, particularly at the early phases. That's when our processes and analytics are not entirely robust. And at tox and phase 1/2 manufacturing, we may need to make some changes readily. And it's important to have our experts close to the manufacturing space. And that's absolutely key.
Okay, good.
And now I would say that the key is building that facility close to our development labs. And in Brisbane, the main manufacturing facilities on the first floor and development labs are on the 5th floor. While in Valbonne, France, and that's where our cell therapy facility is, our manufacturing space is just across the street from the development labs.
Okay, excellent. So certainly geographic proximity matters quite a bit. Now, of course, you mentioned cost, resources. What other major considerations did you have when building the facility?
Well, given that this is, again, I was talking about these are early phase. The process is still being developed, and so it's really important that you have these facilities as flexible as possible. So our rooms have no fixed equipment. Essentially, they're small ballrooms with utility panels to hook up the necessary equipment for a certain manufacturing campaign. We rely heavily on disposables, so we cut down our water use, and also the need for equipment for cleaning and sterilization, which adds cost and also makes the facility a little bit less flexible. And I do think that this design had helped us make a change that happened after we actually designed it and start to build the facility. We had a fill finish suite. We decided since the air quality for a fill finish suite is very similar to open processing cell therapy suite, we needed that functionality. We didn't have it because it was designed for AAV, but we were able to move that area, make it a cell therapy suite. It's a very similar footprint, but the equipment's different. But it's just, in the end, cell therapy actually is mainly all benchtop equipment, and we're able to get that in pretty easily. And then what we did is moved our fill finish operators using isolator technology to another suite that had lower air quality requirements. So again, we've already able to see that the advantages of that flexibility to allow us to make these changes.
Good. Okay. Let's expand on that a little bit more with where we are today. So that decision to build your own facility, how has it affected the company's ability to move forward from, whether it's, cell therapies, gene therapies, from early stage to actually delivering treatments?
Well, it is early in the process here, and it's early stage. It takes a while to get from phase 1, 2 to phase 3 in commercial. But I will say that we saw an immediate impact with building this facility. We had some troubles with one of our products at our CMO, and then we decided that's where we basically converted our fill finish suite to cell therapy. We transferred a process to that new space in Brisbane. And we were able to do that in 15 months. What we were able to do in 15 months was what our CMO could not do in over 2 years trying, and that's to produce 3 batches in a row at high dose. So in the end, we are able to do the things that, having our experts close to this process. I mean, tell you the truth, one of the issues with early stage at CMOs is they don't want you to have your experts in there tinkering while you're doing a process, and to be able to highlight and manage issues so closely. And in the end, it's really important you have, actually, as part of your manufacturing team, your experts, right? And you need them close. And we saw that value just with this first campaign in our Brisbane facility.
Gotcha. Okay. So that's actually what I wanted to talk through next. A little bit more of the specific challenges you needed to overcome. So certainly it sounds like when you were working with the CMO, one of the things that did hinder you was the fact that they weren't allowing your experts to be part of the whole operation. So, excuse me. What are some of the other manufacturing challenges you needed to overcome as you built your facility?
I mean, the biggest challenge, and that's actually the same issue, probably the biggest issue that a CMO has, is hiring and retaining talent. Once you get the right people and they are trained and able to do the operation, when it's a very complex operations, particularly for cell therapy, you don't want to lose them. And so it's very important to train them. And that was one of the issues with the CMO we were having. Now, Brisbane is in the middle of the San Francisco Bay Area, right? And there are a lot of companies with similar needs, similar staffing needs. And that's great that you have that pool of people to hire, but at the same time, on the flip side, we are also, we could also have that issue where people will take our staff, and often they will be wooed away with promotions and more pay, particularly in the Bay Area. And so we had to rely on other means to retain our talent. And I think our collaborative culture was very good. We really care about our staff and have fabulous leadership. And I think overall we have some really smart and resourceful people. But yeah, that's been the challenge. But I think we've had some issues with that. But in the end, the last 18 months, it's definitely, I think we're in a definitely better place.
Yeah, for sure. And I think that's definitely a topic we hear broadly, not just in manufacturing, but just in all of cell and gene therapy. The need to attract and retain top talent, and how do you do that? And um, so certainly not something that we do. Certainly something we hear quite often. Certainly in manufacturing, but specifically in terms of the whole sector as a whole. Um, Now, speaking of the whole sector as a whole, let's talk broadly about manufacturing. Uh, we know that the cost of manufacturing is very high, very staggering. From your perspective, what are the top challenges when it comes to AAV manufacturing? How is it possible to drive down costs?
Well, for AAV, bioreactor titer and final product potency are key. And we do need to move from adherent to a suspension culture and develop high expressing cell lines. And the industry has used cell lines that require what we call transient transfection to make AAV. And that is inherently inefficient. Ideally, we'd like to move to producer cell lines like used to make monoclonal antibodies from CHO, that's Chinese hamster ovary cells. And that took time to develop that. And part of the problem is it's very important that we move quickly to our products and demonstrate something that's of value, right? And then cell and gene therapy is very early. It takes years to develop a good producer cell line. That extra time, for a lot of the smart startup companies can't afford that extra time. But it's something that will evolve over time. And I think eventually, as we get better at this, there will be producer cell lines that will allow us to move these products more readily and at lower costs into the clinic. And then the other thing is, the more potent is your product, the lower the dose needed. And so obviously you need to develop as potent as a product as you can. We use SF9 insect cell culture, which is inherently less potent than the human, the ones that come from the HEK293 and other systems. And I'll talk about that. But in the end, we've had to spend a fair amount of time to improve our potency of our products, particularly the ones in SF9.
Okay, sure. And feel free to continue to elaborate on that. You said you would continue to talk on that. Feel free to.
Yeah, sure. No, well, so in terms of driving down those manufacturing costs, we certainly evaluated better cell lines. We're talking to other companies that focus on that. We've also improved our production processes, and we've designed better constructs using genetic engineering to make our AAVs more potent, and had some success with that. And we're also looking at, I think there's a number of people in the industry that feel that making a AAV from a human cell line will eventually give you a better product. But the problem is the HEK293 has had challenges getting the amount of, basically the titers are just very, very low. So you have this trade-off between having having a high potent product, but then very low titers. At some point, then you might need, so the issue is you want to reduce, as you guys know, one of the issues that we're concerned about is we don't want to have too much AAV. There's toxicity there. So finding that balance, particularly, I think it is going to be important as we drive to get a higher potency product, because then we don't want to have our titer as high. So that is something that we're striving to do as well. And we're also looking at going back to human cell lines for that, but it will require a fair amount of effort to drive the titers to get them up to the levels that we're seeing at, say, SF9 insect cell culture.
Okay, good. Thank you. Thank you. Any commentary, though, on just from a cell therapy perspective, manufacturing there? Is there anything we want to address
Yeah,
in terms of cost?
obviously for autologous cell therapy, you have each run is for one patient, and that one run could cost several $100,000, right? And that is not feasible for our healthcare system. We need ways to address it. The key to doing that for autologous is to close and automate the process. And that way you can make more patient preps within the same facility, the same space, and the same staff. Now, the holy grail for cell therapy is moving to allogeneic, where one batch can treat dozens or hundreds of patients. And we use our zinc fingers nucleases to edit cells to make them allogeneic. And we're very excited about the prospect of getting to that point.
Good. Okay. So for the companies out there listening to this who are considering building their own in-house facility, what advice do you have for them? They may not know what they don't know. What is their first move if they are struggling with cost or just any part of it? What would you say are the top tips, best practices for moving forward with even the consideration of building your own facility?
Yeah, so for AAV, you do need to have a path to move to a higher titer, hopefully a suspension cell line, and ideally do that before you get to tox in phase 1-2. You can make that change from phase 1-2 to phase 3, but I tell you, it's getting harder to make that switch from a comparability standpoint. So from that end, obviously, and this is something even I know from my days working with CHO cell lines, right? It's basically ideally, if you're going to make a cell line change, do that before phase 1-2. It just has a much lower hurdle from a regulatory perspective. For cell therapies, you certainly need to have a path by phase 3 to move to a closed and automated system. It is just it's challenging. It's going to be costly in the clinical phase, just because getting slots at manufacturing at a CMO, just to manufacture is going to be a couple of $100,000. But you need to find a way to drive that down definitely for phase 3 commercial, because in the end, it's going to be harder to justify that cost, I think, long term.
Definitely. It makes a lot of sense. Thank you. Okay. Well, we have reached the end of the formal end of our episode. And at the end of every episode, I like to ask my guests who they are when they aren't in the lab or the office, to get a little bit of an idea of who they are. And so I know you're in the Bay Area in San Francisco, and just some ideas for someone who's never been there. Now, Golden Gate Bridge and Alcatraz Island aside, what would you say are the non-tourist stops that are a definite can't miss for anyone visiting the San Francisco Bay Area?
Well, I'm an avid outdoorsman, and I'd kind of point to those places to visit. I'm actually a bay swimmer, so I swim, and the water's like 50 degrees right now, so it's pretty cold, but once you get used to it, it's quite exhilarating, and I get a lot of enjoyment hanging out near Aquatic Park next to Ghirardelli Square. Hyde Street Pier has several older and massive sailing boats. The Balclutha at the end of Hyde Street Pier is definitely a sight to see. Definitely sights to see. I also live in Marin County, and I mountain bike above the Golden Gate. And from there, you have a beautiful view of the city and the Golden Gate. So going up there, even without a mountain bike, it's pretty spectacular. And I also do a fair amount of hiking and biking near Mount Tam. And it's just the most spectacular scenery there. And Marin is where mountain biking was first developed. And yeah, it's just, like I said, it's amazing fire trails, and people can rent bikes to go up there. It's amazing. And also to encourage anyone to hike in Muir Woods. There's a national park there.
Nice. That's quite a fun fact too about the biking, that it was originated there in that area. That's very neat. How often do you swim?
I'm in the pool a couple of times a week and in the bay a couple of times a week, but it is one of my main, it definitely keeps me sane.
Right. I always say that the mental health benefits are equally, if not even better than the physical health benefits when whether you're biking or swimming in the bay, or any form of fresh air will do it for sure.
Yeah, no, in this industry where you're under a fair amount of pressure, it's good to have
High stress.
Yeah, exactly.
Absolutely. Well, thank you for that. That's quite a breakdown of all the things to do if you're in the Bay Area or plan to visit it anytime soon. So thank you. All right, that's it for this episode of Cell and Gene, the podcast. Thanks again to Sangamo's Andy Ramelmeier for his time and insight. Andy, this was a lot of fun. Thank you so much.
Okay, great. Thank you.
Be sure to visit Cell and Gene regularly, subscribe to this podcast, and share it with your colleagues. Thanks for tuning in, and we'll talk to you soon.